Okay, let's get started. So welcome everyone to the Allen Institute and our modeling software workshop. I'm Anton Arkhipov. Nice to meet you all again. And I'll give you a quick introduction and then we'll jump right in to our busy schedule. So first things first, I want to thank the team for preparing everything. It's been a lot of work. So first and foremost, Kale Dye, our software engineer. So he made essential contributions to developing all our tools and then setting up everything, including AWS accounts and images that you all will be using here. So yeah, thanks a lot, Kale. And then other team members are scientists, Shinya, Daryl, Beatrice, Laura, and Yelena. All of them are there, your nice and friendly guides and TAs. Xiaoping is another scientist here at the Institute who has been helping us. And then you saw her downstairs, Emily Borsom. She's a project manager, so she's done a lot of work behind the scenes and with our education team to make sure everything is set up for us. And then our friends from University of Illinois, Barry, Jason, and Amat, and Barry's here in person. So we are collaborating with them on this project and software development for the community. So yeah, thanks a lot for everyone's efforts. All right, and yeah, with all of you, when we have lunch today, I would love to hear about each of you. We'll have sort of a round of introductions. And yeah, looking forward to spending the time together over the next 3 days. Well, so you all are here probably because you have a dream of doing something interesting, scientific, and perhaps that dream involves modeling the brain or some components of it. And we hope we can help. So what are we going to be learning these 3 days? These are the tools that we developed here and share with the community, and thanks to the generous support from the NIH through this grant, U24 grant, we can continue development of these tools and support dissemination. And yeah, this workshop is a part of that, so thanks very much to the NIH for doing this. We have 3 tools that we offer. One is called Brain Modeling Toolkit, or BMTK. So that's the tool for building models and running simulations of models. Then the one in the middle here is the Sonata format. So it's a file standard and format for sharing models and exchanging them in a standardized way. And then the third one is Visual Neuronal Dynamics, or VND. And that's a tool for visualization, for 3D visualization of models and simulations. So let me go through each of them in a little bit more detail. So Brain Modeling Toolkit, what is it? It's a API, Python package, which has multiple modules. One of the modules is Model Builder, so it helps you to build your models. And then other modules have to do with running simulations at different levels of resolutions. And how we make it work is essentially we leverage existing other tools that are very powerful for simulations, such as Neuron or Nest, but we provide a sort of a unified interface for working with them through BMTK. So we are not trying to reinvent the wheel, we're just sort of trying to make it easy for you to work with the tools like Neuron and Nest, which are very powerful, but to be, to really take advantage of them. In principle, you should become familiar with their scripting languages, with the details of how they work, and it's very complicated. And BMTK does a lot of that job for you. So, in principle, you can run fairly sophisticated simulations with little to no coding, and with a very similar approach and interface between different levels of resolution. But if you want to become, more advanced and do coding with BMTK, you certainly can. It's open source software. You can, in fact, contribute your own code. And so, yeah, it can become pretty involved if you want to. So, okay, then how is this all possible that you can work with very complex simulations with little to no coding. So one really important aspect of it is this file format Sonata, which allows us to represent models and simulation in a standardized and computationally efficient way. So we developed this format together with several other teams, published in 2020, shared with the community. It's as everything else is open source. I think there might be an extra chairs there in that closet. So it contains all kinds of information for representing models and simulations. So if you need, what do you need for models? You need to know where your cells are or the nodes in your neural network, what their properties are, who they are connected to, what the properties of connections are. Then you need to know, how long do I need to run my simulation and all these sort of parameters, maybe a temperature of your simulation. And then finally, when the simulation is run, you get the output, you get the spike times, you get time series, right? And so all of that can be represented in Sonata format. It's also pretty flexible. It has some reserved attributes and some optional attributes or even user-created attributes. You can put some of the data into human-readable text formats and other more heavy data into binary formats. So a lot of flexibility, a lot of computational efficiency, and we are sharing our models using this format with everyone, and some other groups do as well. And it's not only our tool chain that works with this model. It's actually, with this file format, it's actually a pretty substantial ecosystem. So here's just a list of different tools which allow you to create models in the Sonata format or to read them, simulate them, visualize them. So quite a bit of coverage. Okay, and then last but not the least is the visual neuronal dynamics. And so this is the tool developed by the team at University of Illinois, with whom we are collaborating. And it's a tool for 3D visualization of models and simulations. You can create images, you can create movies. You can work with it on your laptop. You can work with it on specialized hardware. It's quite versatile and highly efficient. All right, so these are the three tools that we'll be teaching over these 3 days. I mean, if you look at the tutorials, they actually primarily focus on BMTK, on model building and simulation, but we also have something about VND, and Sonata is sort of there to support everything. Now, what I would like to do, yeah?
Just a quick question. Are you going to make these PowerPoint slides available afterwards?
Yeah, we can. Yes, sure. We'll do that.
Thanks.
Sounds good. Yep. Okay, so now what I want to do is to talk about some scientific applications. So you may ask, okay, good modeling, but what is it all good for? And of course, you can do a lot of things with it, but I want to illustrate to you what we do with these tools, and really it all emerged from our scientific needs. We developed these tools because we needed them for our own work, and then we realized that others might benefit from them as well, and so we share them with the community. But for us, what we are trying to develop in my group is are models of brain circuits and ultimately of the whole brains, which are highly biologically realistic and systematically informed by the data. So being here at the Allen Institute, this is really great because we have a lot of data. And originally, the Allen Institute was sort of known for the things like gene expression atlases, but in the recent decade we've had a systematic program to obtain many more types of data, and so we have a lot of data about the composition of brain circuits, so the cell types, their transcriptomics, their morphologies, their electrophysiological properties. So these are building blocks of your circuits. We have a lot of information about their connectivity from electron microscopy connectomics, but also from other approaches such as synaptic physiology, multi-page synaptic physiology, or mesoscale connectivity using viral tracing. We have a lot of data for activity in vivo, so 2P calcium imaging on neuropixels electrophysiology from awake behaving animals. And ultimately, we are trying to put all of this data systematically and carefully into models using the tools that we are talking about. And just a few months ago, a whole bunch of us here at the Allen Institute published this paper that's shown there at the bottom in Nature Neuroscience. It's a perspective paper. So if you are interested about sort of what kind of modalities are covered and available, you are welcome to read it and see more details. All right, so now this is a visualization of our, maybe I can call it a flagship model, a model of mouse primary visual cortex, so V1. We put together a lot of the information about the cell types, their connectivity, activity in vivo, and created this highly comprehensive, detailed model. As you can see, it can take in inputs, the visual stimulus as an input. And that is all highly constrained by data. You can use arbitrary visual stimuli, so any movie that you like can be used as an input to this simulation. And then the model will create activity in the circuit that's reflecting this sort of input. Everything that you see here is created exactly with the tools that we are teaching at the workshop. So the model was built using BMTK, was simulated using BMTK. It's shared with the community in the Sonata file format, and the visualization, the images, the movies are made with VND, so by Barry. So yeah, you can, in principle, once you learn these tools, you can do it all by yourself. So we published these models about 5 years ago. There are actually 2 versions of it. There is the biophysically detailed version with morphologically detailed reconstructions of neurons and Hodgkin-Huxley equations to describe ion channels. There is also a version that's a point neuron version, where we have a mapping between the biophysical and point neuron version, neuron by neuron and connection by connection. Biophysically detailed, of course, is computationally demanding. It takes hundreds of CPUs to run it, and you get maybe 1 simulated second in 1 hour or so. But the point neuron is much more accessible. You can run it even on a single CPU, and you can get 1 simulated second within 3 minutes or so. And now we have this has been developed further, so we can run models like this on a GPU, maybe close to real time. The models are highly detailed, so 250,000 neurons, a lot of different cell classes, a lot of complexity about how they're connected, and so on. And so just really trying to incorporate the realism of real biological data that are collected here and in other sources. And like I said, we can use arbitrary visual stimuli, so you can provide a natural movie or some kind of artificial movie like a drifting grating as an input to this model, and the output will reflect that. Then you can compare statistically the output and the activity of the different cell types that you obtained in these simulations with what is observed experimentally. So yeah, these models and the tools have been around for about a little over 5 years, and we've seen some good uptake in the community, interest from people like you and others, which is great. This plot just shows a number of publications and preprints which came out every year since these models were publicly available, where people did something useful with our tools and models, right? So it's not just the number of citations. It's taking the model or some components of it and applying it for their own purposes, or our tools like BMTK, to do new science, right? And so I think the sum of all of these years is 44 now, which is pretty good. So yeah, I'm excited to see this this working and people using it. And I would like now to talk a little bit about the applications. And so what kind of applications these models can be used for. Just generally speaking, I would say the way I see these models, so they are platforms for discovery, for computational discovery. They are not created for one single goal, to answer one single question. They are created to incorporate a lot of the data, a lot of information to make them biologically realistic, and then you can apply them to many, many different questions. But generally, the types of questions that you ask with these models are structure-function relation questions, right? So how the structure, the architecture of the circuit supports certain function. And for that, we can look, at function as a spike in activity, or on the other hand, we can look at things like brain signals, right? So with these models, at least the biophysically detailed version, you can compute things like extracellular electric field, for example, right? There is a current in each segment of your simulated neuron and each synapse that creates electric field. You can compute the sum of all of that at each time moment. And so from that, you can get extracellular potentials and things like local field potentials and anything like that. So the idea is that you look at your spiking patterns or at brain signals like LFP, and you ask, how is the architecture of my model supporting or making it possible for this particular pattern of activity to develop? And we published a number of papers on that, and many other people have done as well. And now I'll go through a number of slides and tell you, just sort of advertise the things that team members here will be telling you about over the next over the next 3 days. This is all work in progress, cutting edge, unpublished, and I hope you enjoy it. And the first thing that I need to tell you is that we are now sort of in the middle of essentially a revolution in what we are doing. So what I showed you so far is that we can take a lot of the data, and a lot of really systematic data is now being obtained by the Allen Institute and other places, and that's great. But that's been going on for several years now. What has happened in the last couple of years in our field is that we now can use sort of deep learning tools, so gradient descent, backpropagation through time, to optimize our models. And this has been a huge, huge problem for this very sophisticated and large network simulations, because you can put the data together, you can build a network, and you still don't have every single parameter captured. And it's actually very difficult to find a regime for the network to behave well, not to explode in an epileptic-like activity, but on the other hand, have enough activity going through the network to perform some kind of useful computations. And so getting the network to that point has been very, very difficult, and for such large networks, we had to do it essentially by hand. But now, including through the work of some people here, we can actually do it automatically. We can, again, use backpropagation through time, gradient descent, and so this one of the talks by Shinya will be exactly about that. So the new tools that we have now to do this, you define a loss function to capture certain pattern of activity in your network, and then all those constraints are taken into account automatically, and then you get from some, spike in activity like here in epoch 1, which is not particularly interesting and probably too weak, to something much more interesting and powerful within, in this particular case, I think it's within half a day or so, for fairly sophisticated and biologically detailed network. In this case, this is a point neuron simulation, and doing this for biophysically detailed simulations is still a bigger challenge. But at least for this very, very large and complex point neuron simulations, we can do it, and it works really nicely. We don't yet have like a real well-developed tutorial to do this, so this is not the major part of the workshop now, but I want you to be aware of this, and this is coming big time. Okay, so we'll talk about that. Another talk by Beatrice will be about also automatic optimization of these point neuron models themselves, right? The building block of our simulations, and then about some ideas how you could make a correspondence between the point neuron models and biophysically detailed models. And if you optimize the point neuron network model, maybe you can actually move to the biophysically detailed one in a systematic way without re-optimizing it. Then Yelena will tell you about optimizing biophysically detailed neuronal models, also using gradient descent, very exciting area. It's a difficult problem, but she's made some good progress there, and yeah, you'll see it's exciting and promising. And then we don't want to be just working with mouse V1 all the time. We have a bigger ambition. We want to go to, like I said, ultimately to the whole brain, different species. And so you will hear about some work in progress from Laura, simulating a whole mouse cortex. 9 million neurons, I think 27 billion synapses or something like that. Huge simulation, and we still need to do a lot of work to make it really nice, but it's working, and we can get some interesting results. And then last but not the least, Daryl will tell you about our efforts to simulate a piece of a human cortex, working with colleagues in University of California, San Francisco, recording in awake humans with neuropixels, and then using that information together with the data here from the Allen Institute about composition of the human cortical circuit, building a model. And in this case, instead of using visual inputs, we are using auditory stimuli. So again, it's very, very exciting to be able to do this. All right, so last thing that I want to say is just a little bit of an orientation about what kind of training materials we have. So if you go to our GitHub page, or when you log into AWS today, you will see a number of different folders there, right? So they are listed here. And so essentially we have three different things here. One is what I would call main tutorials. So this is primarily about BMTK and being able to run simulations of single cells, of networks, build these models, provide visual stimuli. So you can learn all of this in these tutorials that have CH in front of them, meaning chapter 1 to chapter 6. So these are our main tutorials. Then we have optional tutorials. I mean, like, really, it's all up to you what you want to do. You don't have to do anything, or you want to, if you want to work on any particular tutorial, it's up to you where you start. It's probably just going to be easier for you if you go chapter 1 to chapter 6 first. But here's a whole bunch of optional tutorials that looks at specialized topics, and if something is of particular interest to you, please look at it or look at all of it. It's all quite interesting and powerful. And then we have sort of a newer stuff here in the folder exercises. So this is several new things. Sort of, trying out a mini project, and we encourage you to give it a shot. There are, some exciting things here, like providing network inputs to a biophysical neuron models and trying to adjust it so that you obtain sort of desired activity. You can be computing local field potential, and then another one here is running a whole V1 model simulation on a supercomputer via cyber shuttle. So this is, we have a collaboration with people at Georgia Tech. They developed this thing called cyber shuttle, which is a system to allow sort of seamless. We need extra chairs. I think there should be in the closet, either this one or that one. Maybe that closet right there. Hi, Anna. Yeah, thanks. No. Okay, we'll figure it out. Just let me finish. Do you have a question? Oh, there is a chair there. Okay. Yeah, thanks. Yeah, so this is a system that allows, once you enter it, it allows like seamless access to different types of hardware, different types of setups where you might be able to run your simulations, including like NSF supercomputers. So yeah, this can be handy if you want to run really large simulations. So if you're interested in this sort of stuff, give it a shot. Let us know how it works for you. And then, the last thing here is listed the TensorFlow optimization of a network model, and that's exactly what I was talking about. The sort of revolutionary advance which allows us to optimize these very complex networks not by hand but automatically. Therefore, 5 chili peppers for hot. It's not spicy, it's just really hot and exciting. All right. Okay. What's going on? Is it an earthquake? Well, that's all on my part. So let's stop here. And next up will be a presentation by Kale, who will orient you to how to get started with all the training materials and such. But before that, just quickly, we have a newsletter. If you are not on it, I really encourage you to sign up. We are not going to spam it, spam you. It's only 3 or 4 times a year that we send out some useful news and such. So I encourage you to do that. And then we will have a survey on Wednesday. There is like a special time on the schedule where we ask everyone to fill out survey, and we will really, really appreciate it. The NIH likes us to have this survey, and so that information will be super useful. So it only takes 10-15 minutes and will be very helpful. All right, thanks. Before we switch, any quick questions for me?